Next, let's understand gene mutations. What is a gene mutation? A gene mutation is a change in the DNA sequence. Simply put, it is different from the original DNA sequence. It could be a point mutation, or it could be a segment of DNA added, or a segment of DNA lost. These gene mutations may lead to changes in the function of proteins. It could enhance the function, or it could result in the loss of function. Let's illustrate with a simple example. For instance, a tumor suppressor gene experiences a mutation, then it loses its tumor suppressing function. We specifically call this loss of function. If it's an oncogene and it mutates, its cancer-causing function is enhanced. We call this gain of function. Next, let's look at the types of gene mutations. First, what is a point mutation? It is a change involving only a single nucleotide, known as a point mutation. Gene mutations may involve the insertion of a segment of DNA or the loss of a segment of DNA. Whether it's an insertion or a loss of DNA, it may lead to some displacement phenomena in the gene. These displacement phenomena are related to leukemia. Furthermore, it may also occur during transposition, where a segment of gene A moves into gene B. We call this gene recombination. Regardless of the types of gene mutations, they may lead to changes in protein function. The reasons for gene mutations could be natural mutations or environmental factors, and also inheritance. Now let's discuss natural mutations. Students might wonder, how can my cells mutate on their own? How does their DNA mutate by itself? The reason is that errors may occur during DNA replication, or there might be malfunctions in the repair systems within your cells. It's like having my own immune system, which could originally overcome diseases, but then my immunity weakens. When my repair mechanisms fail, it could lead to natural mutations. Moreover, students, when you are under significant stress, this can also lead to an increase in reactive oxygen species. These reactive oxygen species may attack our DNA. Thus, these mutations may be naturally generated. Now, what about mutations caused by external factors, for example, the carcinogens we mentioned earlier? Next, we will also introduce viruses that can lead to gene mutations. Some inheritance is passed from parents to offspring. Regardless of the reasons for gene mutations, these gene mutations ultimately will be repaired by the repair systems in our cells. This will also be discussed in future units. Next, let's examine the impacts of gene mutations. They may affect our phenotypes and the variations in individual expressions. For instance, the pigmentation of a mole, the size of the mole, its shape, and even may lead to certain hereditary diseases or increase the risk of some genetic diseases. Some of these mutations can affect organisms' adaptability and survival capabilities. Returning to humans, natural mutations are non-mutagens, as we previously discussed regarding induced mutations. Errors may occur during DNA replication. These natural mutations, after some repair, are referred to as the background mutation rate. This is the background mutation rate. You, I, and others, each of our background mutation rates may not be the same. Next, let's look at induced mutations. These are induced by mutagens. These can alter DNA and may also be related to environmental factors. The DNA in cells may undergo changes due to either natural or induced mutations. Changes in DNA sequences that involve point mutations are changes of a single base. They can be categorized into transitions, transversions, and mispairing. Let's first discuss mispairing. We know that when DNA base pairs, A pairs with T. If A pairs with C instead, which was originally paired with T, this constitutes a mispairing. This is a type of point mutation. Next, I will use this example to explain what transitions and transversions are. As you can see, I originally had AT pairing, which becomes TA pairing. The first position A pairs with the first position T in the image. One is a purine and the other is a pyrimidine. It changes from purine to pyrimidine. We call this transversion. AT becomes GC. The first position is A, which is a purine. The first position G is also a purine. Both change to purine, and we call this transition. From the examples of 1 and 2, we can clearly see that 2 is a transition example. It changes from purine A to purine G. Students can also double-check the second position T. It is a pyrimidine. After the mutation, it becomes C. It is also a pyrimidine. Mutating to pyrimidine, we call this transition. When a genetic material gains a segment of DNA, this is related to transposons. Transposons, also known as jumping genes, refer to short segments of DNA that can jump into another DNA. Especially in corn chromosomes, it can be seen that it can jump from chromosomal DNA to plasmids via transposons. Its inherited traits remain unchanged. It may jump from gene A to gene B. It's like having variegated corn. Sometimes you see white and purple kernels, which may be related to these transposons. Typically, most point mutations are predominantly transitions. Do you remember what a transition is? It involves purines changing to purines or pyrimidines changing to pyrimidines. Let's look at this example in the image. Earlier, we mentioned nitrites or nitrosamines, which metabolize in cells. They are classified as group 1 carcinogens. Why are they classified as group 1 carcinogens? They convert our cytosine into uracil. Uracil, during DNA replication, opens the double helix. This is referred to as semi-conservative replication. This means that one strand is retained, while the other strand is newly synthesized. In the example shown, G and C are normal pairings. If I consume too many nitrites metabolizing into nitrosamines, they enter my cells, deaminating my C into U. When DNA replicates, the double helix opens, where U pairs with A, C still pairs with G. However, your UA pairing A will pair with T during the next DNA replication. So my original GC pairing may become AT pairing. Let's check again. G is a purine and A is also a purine. This point mutation, both purines we call transition. Next, let me give another example. This example involves bar-DU-A adenine. The methyl group above is replaced by a bromine atom. When bar-DU is incorporated during DNA replication, bar-DU will shift between keto form and enol form. When it shifts to enol form, its configuration resembles C-C pairs with G. Ultimately, it becomes TA, resulting in CG pairing. Again, the first position is T replaced by C. T and C are both pyrimidines. A changes to G, both purines. This also exemplifies a transition. Next, I want to introduce some terminologies. When these mutations may change our phenotypes, there are several terms to familiarize ourselves with. First, this term is revertance. What are revertants? It means I have experienced a mutation and it returns to its original form. I give an example. If my hair was originally black and I mutated, turning my hair green, we call this a forward mutation. Forward indicates moving forward. If I undergo another mutation, returning my green hair to black hair, this second mutation is called revertance, because it has reverted to my original black hair. Next, another term is similar to returning to the wild type. However, it involves the original gene losing its function and cannot express itself back to normal, which we call back mutation. For example, if I originally had black hair but can no longer grow hair and I become bald, this process of becoming bald is called a forward mutation, as I went from black hair to not growing hair. If I then undergo another mutation and regain my black hair after being bald, this is called a back mutation. Revertance and back mutations both refer to returning to the original black hair. What is the difference between them? The difference is that back mutation originally involved a gene mutation that prevented it from manifesting a phenotype. Revertance may involve differences in severity, for instance, black hair or green hair. I did not become completely bald. This example illustrates what revertants are and what back mutations are. Next, I will explain another term closely related to mutations. This is called second site reversion. The term second site reversion literally means a second mutation that suppresses the effects of the first mutation, which does not necessarily match the original wild type sequence. I will give an example. I have two hands, and the two hands represent wild type phenotypes. Then, I experience a mutation and lose one hand. At this point, this is called a forward mutation, as I went from two hands to one hand. The key point is, if I later receive a robotic arm that restores the functionality of two hands, this is called second site reversion, because I regained the function of both hands through a second mutation, suppressing the effects of the first mutation. Originally, I had a mutation resulting in one hand, but by receiving a robotic arm, I suppressed the effects of the initial mutation. Even though it differs from the original DNA sequence, not being my original hand, I now have a robotic arm. This scenario is termed suppression. If I received a hand back after having lost one, and it turned out to be my original hand, we call this true reversion. So true reversion and second site reversion are somewhat different. True reversion means my original sequence has returned exactly the same, which is termed true reversion. Second site reversion refers to the original sequence not necessarily being identical, but I still have the original protein function, which we call second site reversion. Why does second site reversion occur? This relates to the wobble hypothesis. When our tRNA binds to our mRNA, there is a third base redundancy. This means the third base pair does not adhere to Watson and Crick's rules. This is referred to as the wobble hypothesis. Using this diagram as an example, my DNA sequence is all UUU, which translates to the amino acid Phe. However, the third sequence can be UUC, which can also encode for the amino acid Phe. That is to say, the same amino acid can be encoded by UUUU or UUC. Thus, even if your DNA sequence differs, you may still exhibit the same phenotype. This exemplifies the wobble hypothesis. Students, have you ever wondered if a segment of DNA undergoes mutation? Is the mutation rate for every amino acid in the DNA the same? The answer is no. Each mutation has its so-called hotspot. What is a hotspot? It refers to a specific point within the DNA where the mutation rate is particularly high, which we call a hotspot. Usually this relates to nucleosides modified beyond ATGC. Typically, for instance, cytosine, when methylated, is very likely to become a hotspot. Students, have you ever thought when you go to a restaurant, you open the menu, and there is a section marked as hot? What does that indicate? It means that dish is the most ordered by customers. It's a similar concept. Earlier, I mentioned that cigarettes contain many carcinogens. How did scientists initially determine that smoking is related to lung cancer? They conducted experiments and found that smokers have a hotspot mutation rate for a carcinogenic gene called P53, which aligns with patients with lung cancer. Thus, they indirectly confirmed that smoking is indeed closely related to lung cancer. Next, when these genes undergo mutations, their protein functions will also change. If a segment of DNA undergoes deletion, the protein will not be produced. We call this a null mutation. The protein function is completely lost. If some point mutations occur that change the function or amount of this protein, these mutations are called leaky mutations. If this DNA mutates, but its protein sequence does not change, we call this a silent mutation. Do you remember that the teacher mentioned the so-called wobble hypothesis? It also refers to UUU or UUC, which both encode the same amino acid. Therefore, if the mutation occurs at the third codon, which is the third pairing in the wobble hypothesis, it may result in a silent mutation where the protein sequence remains identical, yet the DNA has indeed mutated. What is a neutral substitution? This means that the protein's functional activity is not affected despite the DNA mutation. My protein originally carried a negative charge, and after mutation, it still has a negatively charged amino acid, which we call a neutral substitution. Today, we introduce the concept of the origin of cancer, which may be related to gene mutations. The forms of mutations may occur naturally or be caused by mutagens. Do you remember which substances are classified as primary carcinogens? In subsequent units, we will introduce these. Within cells, some genes are oncogenes, while others are tumor suppressor genes. After the mutations mentioned earlier occur in these oncogenes or tumor suppressor genes, they may lose their function, which we call loss of function. In contrast, some functions may become stronger, which we refer to as gain of function. The mutations in these oncogenes or tumor suppressor genes are closely related to cancer. Please stay tuned.